And what we found was that, wow, we had a dramatic increase in cellular life extension, and that was dose dependent.
And so set up the psilocybin study for us. What's the basic experimental setup for the in vitro work you did? You're looking at cellular aging. So you're looking at cells in a dish. What are the cells you're looking at? And how do you, just in general, how does one look at this type of thing in this field?
Yeah, so we didn't want to reinvent the wheel. We wanted to kind of use the tools that we have. And one of the tools that we have is these cellular models of aging. So there are different ways to study cellular aging. One of them is this cellular model of replicative senescence. One thing we know about cells is that they can only divide so many times. And so cells have a normal natural life cycle where they are dividing rapidly, and over time they just, they divide a finite number of times until they stop dividing, they become senescent, and then eventually die. And so we can actually follow that cellular life cycle. And so the experiment was really simple. It was taking psilocin, which is the active metabolite of psilocybin, which is what is when you ingest psilocybin, it's broken down into psilocin, and that's really systemically what your cells in your body are seeing. And so we treated cells with vehicle or psilocin over the course of that life cycle. And it was, again, it was a simple experiment. It was like, if you give it, we're going to watch cells as they normally go through this life cycle, and we're going to treat them with psilocin or vehicle and just watch what happens to their cellular life cycle. And what we found was that, wow, we had a dramatic increase in cellular life extension, and that was dose dependent. So meaning the more psilocin we gave, the greater the life extension. And we did that in lung fibroblasts at first, and then we validated that in skin fibroblasts. So we showed in two different cell types that we were able to have this very similar dose-dependent effect. And again, yeah, we saw in two different cell types that we had this dose-dependent dramatic impact on cellular life extension. I think 10 and 100 micromolar, we saw anywhere from like 29% increase in life extension to like over 50% increase in life extension. I mean, there's no question, like we were like, wow, this is not only significant, but I mean, it's like one of those things where, yeah, sure, we can show significance it's always good to show significance with statistics, of course. But when you can see it visually with your eye, it's
Right, right.
obvious, it's like, yes, we of course back this with statistics, but the effect was very dramatic.
And so psilocybin and psilocin, they're famous in the context of the nervous system for their psychedelic effects. People always talk about the serotonin 2A receptor as being sort of the psilocin receptor. But for nearly any drug like this, they're going to have lots of different things that they interact with in different cell types.
And yeah, to my surprise, actually, most cells in the body express the serotonin receptor.
Mm-hmm.
So yes, fibroblasts do express the serotonin receptor, the same receptor that we know that it interacts with in the brain.
So these cells, so even though we're not talking about neurons here, these cells, and indeed many, many, many cell types beyond neurons, have expressed serotonin receptors of different kinds to different degrees.
Yeah, so we looked at a number of different markers of aging, right? And oxidative stress being one of them. We saw that so the net effect was reduced oxidative stress. And I mean, when I say reduced oxidative stress, I mean oxidative stress levels that were actually significantly lower than even young cells' oxidative stress levels.